Hello, my name is Carmen de Miguel. I'm from the University at Alabama at Birmingham, and I am here today with Dr. Amanda Doran from Vanderbilt University Medical Center. And she is one of the invited speakers here at the hypertension sessions. And her research program is focused on immunity, specifically innate immune cells, and how they are involved in atherosclerosis. So, well, I am so happy to be here with you today, and I wanted to ask you, what is your talk going to be about?
Well, thanks so much for having me. Like you said, my background is mostly in atherosclerosis, and so this is kind of a new foray for me into hypertension. So I'm really grateful to have been invited to share our work. This particular talk that I'll be giving tomorrow is going to focus on how we discovered that angiotensin II can act as a driver of something called innate immune memory or trained immunity in order to promote atherosclerosis. And this was really exciting to us because a lot of my prior work has focused on chronic inflammation and mechanisms of residual risk in atherosclerotic cardiovascular disease. And actually, when I first came to Vanderbilt, starting my faculty position, one of my dear friends from graduate school, we did the MSTP together, Mina Maddour was there, and of course, she's a hypertension expert. Yes. And so we connected over kind of shared ideas and came up with this cool project. So I was really surprised to know that even if you treat hypertension, you still have more than twofold risk of cardiovascular disease. And so that, to me, sounded very similar to a lot of what we've seen in both the basic translational and clinical literature as far as managing cholesterol in atherosclerosis. And so we wondered if maybe there was a similar story there. So we were very interested in these mechanisms of residual cardiovascular risk. Mm-hmm. And so one thing that my lab has been very interested in recent years is this innate immune memory or trained immunity. And so that is this process by which myeloid cells, so macrophages and monocytes, are able to learn and remember exposure to a prior stimulus. And unfortunately, they're not smart like the adaptive immune system. They don't develop these highly tailored responses to particular antigens. But if they get inflamed once, they can undergo metabolic and epigenetic rewiring that then leads to a later ability to mount an augmented secondary response. Unfortunately, it's not specific, so there's some crossover between the types of stimuli that can produce both the training and the re-stimulation. And so that obviously can be a good thing for infectious disease, but it seems like a setup for chronic inflammation, especially in a chronic.
And how do you think that those immune cells get to do this memory kind of work? Do you think, since in your project you had angiotensin II, do you think that that's through 81 or 82 receptors on those immune cells?
So we don't actually. So one of the really shocking things about this project is that, yes, we found that angiotensin II can act as a training stimulus. So just like fungal components, bacterial components can do it, angiotensin II can also do it. So it can prime these progenitor cells, these myeloid cells, to have an augmented later response.
I see.
But it does not seem to be related to blood pressure. It's independent of blood pressure, and it does not seem to signal through either of the angiotensin receptors. Shockingly, what we found is that angiotensin II can bind to something called the dectin-1 receptor. And dectin receptor is actually a fungal receptor. It recognizes beta-glucan, so really has no business interacting with angiotensin II. But it does, and in a site that's distinct from where it binds the fungal antigens. And so through that dectin receptor, which has been implicated in other forms of trained immunity to fungal components, angiotensin II can promote rewiring of the epigenetics and the metabolism.
That is so interesting.
It's fascinating.
And then you said myeloid cells, but you only mentioned monocytes and macrophages. So what about dendritic cells, which have been also involved specifically with hypertension, right?
Yeah, so we have not specifically looked at dendritic cells or neutrophils with this particular phenomenon. We're really kind of just getting into this, but I would guess they probably get trained in a similar way. The really intriguing thing about this angiotensin driven training is that it doesn't just happen on the mature monocytes and macrophages. What we see is that dectin receptors expressed on hematopoietic stem cells, and so angiotensin II can train those hematopoietic stem cells, skew them toward the myelopoietic lineage, and affect everything downstream. So we can take take a mouse model of hypertension, do low-dose angiotensin II for just 2 weeks to expose them, take just the marrow from those guys and put them into completely naive mice. And the mice that were trained by the angiotensin signal and receive that marrow go on to develop more atherosclerosis than their counterparts.
That is fascinating.
So it's transferable. We think it's probably heritable to some level. And it happens very upstream. So you can imagine that that has a lot of impact downstream, because if you've rewired a mature cell, those eventually will turn over. But if you rewire a progenitor cell, every progeny from then on is going to maintain that phenotype.
Yes, that's an important research. Okay, so my last question was regarding sex differences. So we know that specifically inflammation is very different, specifically in hypertension. I'm not sure about atherosclerosis, so you will have to educate me on that. But do you see any differences in sex, like females versus males?
Yes, so that's actually something that we're working on now. So we have currently only done the males in sufficient quantity to understand it, but we've started doing some basic studies, and it does look like there's going to be sex differences. And I'm very excited about that as well, as a reason for possibly explaining why premenopausal women are protected against heart disease. And maybe there's something about their resistance to training compared to the males.
That's very interesting.
Yeah, we'll protect them, but we'll have to see.
Yes.
We have a very talented graduate student working on that project.
Okay, cool. So my last question, sorry, I lied before.
No.
This is my last question. So what do you think that the next steps in this project would be?
Well, we were very excited to find that there's a lot of human relevance to this. So in addition to what I described with the work that we've done in mice, we've been able to get human CD34 positive progenitors and show that they can also be trained the same way in vitro. We've been able to do some population level data where we did a predict scan analysis where you look at genetically predicted expression and show that higher levels of dectin were associated with increased atherosclerotic disease risk. And so we think this is probably very likely something important in humans. And so we're hoping to be able to move this into a population study. We've got some early data at a UK biobank with a soluble fragment of the receptor that's also associated with increased risk of disease. And so we'd like to see if we can use this as a potential biomarker. So we try to take it a little bit translational.
Very interesting. Yeah. Okay, well, thank you so much for meeting with me today and for talking about your research, Dr. Doran. And that's
Thank you for having me.
Thank you.